In this video, we'll be looking at how one single chemical reaction that makes carbonic acid is responsible for creating an amazing Australian landscape. We'll also be looking at how exactly the same chemical reaction mediated by an enzyme, carbonic anhydrase, is responsible for a wide range of important biological processes. Well, we're here in beautiful Robe on the traditional lands of the Boandic people, and as I was walking along the other day here in the campsite, there at my feet, just walking there on the road, I found a beautiful little fossilized shellfish, this beautiful little fossilized shell, and it amazed me. But in truth, it turns out I shouldn't have been quite so amazed, because this is limestone country. The whole countryside here is made up of limestone that was formed at the bottom of a sea somewhere between 15 and 38 million years ago, and it's full of fossilized shellfish. In addition, walking around the coastline, it's been impossible not to notice spectacular erosions in the rock. Amazing pockmarked, worn out, weathered, sometimes honeycombed rocks, which I'd never seen anywhere else before. We saw other features of limestone country, sinkholes. We went swimming in a sinkhole, very cold. The blue lake in Mount Gambier, apparently, one theory is that the blue colour that it has, startling blue colour, might be from the precipitation of some calcium carbonate precipitous in the blue, but no one's really quite sure. We also visited the beautiful Narracourt caves, and in the caves we saw wonderful stalagmites and stalactites, and all of these landforms, all of these things, they form because of the action of carbonic acid. Now when carbon dioxide reacts with water, it produces carbonic acid, H2CO3. Now, the carbonic acid is fairly unstable in water, and it quickly dissociates into bicarbonate ions and hydrogen ions, and I'll talk more about that in a few minutes. But the carbonic acid can react directly with the calcium carbonate in the limestone, and this dissolves it away. And when the solution evaporates, well, it can redeposit the calcium carbonate, and it's this dissolving that gives us the eroded rocks and the caves and the sinkholes, and the deposition that gives us the beautiful stalagmites and stalactites that we're seeing. It's amazing that this one acid has so many interesting manifestations in the landscape. But what also is interesting, I think, is that carbonic acid turns out to be very important in many biological processes, and that's what I'd like to talk about today. But before talking about that, we really need to spend a few minutes speaking briefly about enzymes as a class of proteins. Now, enzymes are a class of protein that all catalyze chemical reactions, multiple different types of chemical reactions. They work by lowering the reaction energy and making chemical reactions much faster than they would be otherwise. They greatly speed up chemical reactions that otherwise would take far too long in order to be of any use to living systems. The way that they all generally work is that substrates, which are the chemicals that the enzymes are going to work upon, they bind to the enzyme to form an enzyme substrate complex. Beautiful bird has just landed over there. I can't show it to you because you're looking this way, but not to worry. They form this enzyme substrate complex, and the enzyme massages this the molecules that it's working on, and it spits out product. It spits out the result of the chemical reaction. Enzymes are critical for pretty much all life processes, and in the human body there are around 75,000 separate enzymes. But what's interesting to know is that many of these 75,000 separate enzymes, they actually have the same chemical reaction. They mediate exactly the same chemical reaction, but they're tweaked and modified from each other in order to work in different locations. And when that's the case, they're called isoenzymes. Isoenzymes, so groups of isoenzymes, all have the same chemical functionality, but they're slightly modified to work in different places. And in fact, one of my interests is isoenzymes. One of the very first things that I did when I was a PhD student, one of the first pieces of research that I published, was a study where I developed a method for identifying different isoenzymes in microscope sections and used it to determine that the isoenzyme of alkaline phosphatase that's in blood vessels is the same as that found in liver, bone and kidney. So I have a sort of personal interest and history in isoenzymes as a sort of subject. One important function or one important property of enzymes is that if there is enough product, if the chemical reaction has gone along to produce enough product, and if enough product starts to build up, it'll slow the reaction down and will actually stop the enzyme from working. And in fact, if there's a vast amount of product, then it will actually drive the chemical reaction in the reverse direction. Now that's an important property that will become important in a moment or two when we're discussing this enzyme that's really the subject of today's talk, and that's carbonic anhydrase. Now this is an enzyme that catalyzes the reaction of carbon dioxide with water to produce carbonic acid. Remember, that's the same chemical reaction and the same acid that's responsible for this amazing landscape. Now there are various isoenzymes of carbonic anhydrase, and they have many different functions. They control blood and tissue pH. They control water and electrolyte balance. In the stomach, they produce the acid that makes the acid of the stomach do its magic, do its work in digestion. Bone is a mineralized tissue, and it's constantly changing shape in response to the forces that are applied to it. Well, the cells that resorb bone, osteoclasts, well, they have to make acid to remove the mineralized part of bone, and once again, it's carbonic acid that does that job. White blood cells chase down bacteria and phagocytose them, eat the bacteria and break them down. Well, they produce acid to assist in this breakdown process. Again, carbonic anhydrase does that job. The thing that's remarkable really is that we even need carbonic acid and carbonic anhydrase to breathe. Now you probably know that haemoglobin transports oxygen, that it picks up oxygen in the lungs and it transports the oxygen in red blood cells round to the rest of the body. What you might not know about is something called the Bohr effect, named after the clever scientist who discovered it many years ago. Now it turns out that acid, hydrogen ions from acid, that's what really an acid is, something that produces lots of hydrogen ions. The hydrogen ions bind to haemoglobin, and when that happens, the haemoglobin molecule flips into a new shape. In this new shape, the strength of binding that haemoglobin has for oxygen is reduced. This strength of binding is reduced due to this binding of hydrogen ions, and that type of shape change in a protein is called an allosteric effect. It's very common in biology. Many proteins have interesting allosteric effects like that. Now, tissues make carbon dioxide as a metabolic waste product. When we metabolize food products, I suppose, glucose and fats and things, it's generating carbon dioxide. And this carbon dioxide is a waste product in the tissues that we've got to get rid of. Well, the carbonic anhydrase that sits inside of red blood cells actually metabolizes, or I shouldn't say, catalyzes the reaction, converting this carbon dioxide into hydrogen ions and bicarbonate. So the carbon dioxide that's in the tissues is being converted into hydrogen ions and bicarbonate molecules. Now these hydrogen ions now are available to bind to the haemoglobin, and that flips the shape of the haemoglobin, and now in this flipped shape the haemoglobin doesn't grasp the oxygen so tightly, and so it starts to release the oxygen into the tissues where the oxygen is needed. Meanwhile, as said, the carbon dioxide is also being converted to bicarbonate molecules. Now these bicarbonate ions will tend to shuttle out of the red blood cells into the plasma, and in this time, of course, the red blood cells and the plasma is being pumped out of the peripheral tissues up into the lungs, where the pH is higher. That is, that there are fewer hydrogen ions. out in the lungs. It's a higher pH. Now, it turns out, of course, now in this environment, the haemoglobin molecules can release their hydrogen ions, and this has two effects. By releasing the hydrogen ions from the haemoglobin up in the lungs, the haemoglobin now is able to flip into its new shape that is very hungry for oxygen, that binds oxygen very tightly, and it can now pick up oxygen and load up with oxygen, ready to go out to the tissues again. But also, the hydrogen ions that have been released from the haemoglobin are now available to react with the bicarbonate that's in the plasma. The bicarbonate, that's really the solution form of most of the carbon dioxide that's being carried from the body, from the tissues. This produces carbonic acid. And the carbonic anhydrase, again now in the lungs, in the red blood cells, can take this carbonic acid and do the reverse chemical reaction and convert the carbonic acid into carbon dioxide and water that you can now breathe out. You can breathe the carbon dioxide out of your lungs. So the same chemical reaction that's mediated by carbonic acid that produces the landscape around us is also involved with breathing. And indeed, the same reactions are mediated in protecting us, interestingly enough, from dental decay. Coming from dentistry, I just have to talk about this. Now teeth are mineralized tissues. They're largely calcium hydroxyapatite, and dental decay is driven in large part by acids that are produced by bacteria in the dental plaque as a waste product. So acid is a waste product from bacteria metabolizing the stuff that's in the plaque, and that's what's causing the decay. Now, there's an isoenzyme of carbonic anhydrase in the salivary glands, in the secretory cells of the salivary glands, that generates a bicarbonate, and this bicarbonate goes into the saliva and it flows out into the saliva, into the mouth, and there, in the mouth, it's very important in helping to mop up the acid, the hydrogen ions that are produced by bacteria, and therefore removing or reducing the amount of acid that's available to cause decay. So the carbonic anhydrase that sits inside of the salivary gland has an important role in helping to produce or in helping to protect us from acid made by decay causing bacteria. But in addition, the salivary glands also produce a separate isoenzyme of carbonic anhydrase that's released into the saliva itself. It turns out that about 3% of the total protein in saliva is this particular isoenzyme of carbonic anhydrase. Well, that catalyzes then the reverse reaction, producing harmless carbon dioxide and water. This is a histogram from a study that showed that people that have high levels of this isoenzyme of carbonic anhydrase in their saliva, well, they have a low DMFT, decayed, missing and filled teeth quotient. So that DMFT is just a measure of total sort of decay. It's sort of a group measure for the amount of decay that someone's got. So that those people who have high levels or had high levels of carbonic anhydrase in the saliva, they were protected from decay, whereas those who had low levels of carbonic anhydrase in their saliva, they tended to have more decay. So the same chemical reaction that gave us this fabulous landscape and the beautiful caves that we explored the other day is important for controlling acid-base levels in the body and water and electrolyte balance and digestion and bone resorption, helps us to breathe and even protects us from dental decay. And somehow for me it seems like a wonderful example of how evolution tends to tinker with whatever happens to be to hand. In this case, the chemistry of carbonic acid. So look, I hope you've found that interesting. If you have, then please do like and subscribe and share. And even if you don't, I'd like to thank you for your attention.